Good. Great. Thank you, everybody, for joining. We're really excited to have Manny with us here today. And just as a brief background, he was born and raised in Puerto Rico, did his PhD at University of Chicago, and is currently a postdoc in Peter Sudmant's lab, studying the evolution and longevity in long-lived mammals, especially bats and whales. And he's done work as like a wet lab biologist, doing field work, computational methods as well as several DEI and outreach efforts. And so interested in any of those, he'd be the person to talk to. And with that, we're really excited to hear his talk on pipelines.
Let me say, okay with the meeting being recorded. Cool. So I guess the first thing I want to ask is, does everyone have a test data set that they want to use for this session? Just raise your hand if you don't. Cool. I'll share one with you on flash drive then, when we get to the interactive parts, and then you can kind of follow along using that one. It'll be convenient because then you'll be following along exactly what I'm doing. So today, this is kind of a semi-informal talk. So I'm going to be showing you guys what I've done so far with creating these reproducible pipelines for making manuscripts. And as an agenda for what today's going to look like, I'm going to really be starting about what even is a reproducible manuscript and why do you care about it, some examples and the tools that I tend to use for these things, and then we'll work together to show actually build your first reproducible website and manuscript. So probably preaching to the choir here at this point, but I think the biggest issue right now and the biggest motivation behind all this is the fact that there's this ongoing reproducibility crisis. So just as one example of many papers have come out on the topic, in 2016, Nature did this survey of a few thousand scientists to see, just asking some basic questions about have you ever tried to reproduce someone else's research? And the results of the survey were pretty stark. 70% of scientists fail to reproduce either someone else's work or their own work. When they're looking back through the literature and looking at published results. That's kind of bad. It's pretty goddamn bad, actually. And so in general, this was originally a big thing for the psychologists, in which case everyone kind of pointed at psychologists and laughed at them. But it became pretty apparent when this survey came out that it's not just psychologists. Actually, this is something that affects all scientists across the board, right? If you think about what this actually means in brass tacks, right? So we're not strangers here to how science works. You do science, there's magic, you get data, there's you focus on the data, try to get results, you get the manuscript, you repeat that last process until everyone in the room is happy somehow, or at least equally unhappy, and eventually you end up with a publication. And so we could have our own talk on the reproducibility crisis in the actual brass tacks scientific methods, but that's not what this is for. And when we're talking about data and results aspect of things, you should have come to Stacy's talk last week, last month, where they went over how to use Snake, Make, Mamba, Biocon, and different pipeline tools you can use in order to make your automate and streamline the data to results pipeline.
That's recorded on the website. We weren't there.
And there's a very convenient repo as well. So my portion of this is really focusing on the results to manuscript and even to the publication aspect of things. So basically all the stuff that comes after you have a general idea how to go from data to results. So if you think about what it takes to have a reproducible manuscript, in other words, a manuscript that you can go from the data all the way, So in other words, a reproducible manuscript is one wherein you can go from this data all the way to publication, in n number of times, and you get the exact same result every single time. And so that would be the gold standard is I just click on a button and I have your paper. I didn't need to download anything off of a journal. I literally went to GitHub and I got everything and I made your paper. So in terms of different categories of reproducible manuscripts, the kind of bronze tier, you got a gold star for doing the least amount of work possible. You put your code on GitHub and you made your data available somehow. And actually, it's kind of funny to look to see how much variability there is just on this alone. There's GitHub and GitLab, but then there's also just some I don't know zip tarball in the publisher, but then the publisher goes out of business, and so that tarball disappears. That happened to me in the process of doing reproducibility stuff, where it's sometimes like, you'd be surprised at how poor, or not surprised, how poor of a job the publishers do in maintaining all this stuff, which is why we now have a healthy ecosystem of other tools like GitHub, but also like more like open source and not like commercial like versions like Data Dryad, Zenodo, and like Figshare, where the goal of these services really is to be able to have your data up and also have like a DOI tracking that version of your data that then you can cite specifically in your study, and then like everyone can make sure that they always use the exact same code and the exact same data. Now then, that's kind of like the best parts of like, the bronze tier, but really what we're looking for is like a little bit more, right? So like if you take the next step in making your manuscripts more reproducible, you can do the stuff that we talked about to make your pipelines for analysis more automated. But you can also do things wherein you make the pipelines to make the figures semi-automated. And so then finally, you can also make sure that not only your data, but your metadata is available, and you make them sure that they're all available in standardized open source formats. So this kind of depends, right? So like for PopGen, like VCF is like, like you don't even think about it. You're already using a pretty standardized like open source format. If any of you were analyzing data like flow cytometry, for example, or microscopy data, like just having like, the general like TIFFs or your FCS files, like these files that like everyone's kind of agreed in the field, this is what it's going to be looking like, and you're not using the manufacturer's special proprietary version, right? There's like levels to this. Like, sure, you can start with like the automation using SnakeMake, but ideally in the gold star, if you do this part good enough, you can actually make everything fit on a Docker image, so that the Docker image just builds everything for you. You can use like these continuous development, continuous integration methods from like CompSci like Jenkins, and also GitHub has their own version as well, so that you upload tests along with your like data, right? Everyone here has like gone through like the nerve-wracking like trying to get a function to like analyze your data, right, and analyze the test data set the right way, right? The only difference between you and someone who has a job in high-paying CS field right now, other than the fact that you chose to be in this room and not the high CS fields, is the fact that they will take that bit of code and make a test out of it, that then every time they put a commit onto GitHub, it actually runs that test and makes sure that you didn't break something, right? And so I don't know how many of you can relate to this, that in like 3 a.m. in the morning, you're trying to figure out like how to do something like for a conference. You like make all these changes, like your code works, it's great. You go to the conference, you present, you go back, and then suddenly everything else that wasn't what you were presenting on the conference is like absolutely smattered. So like you can't do anything anymore. That is one way of avoiding that from happening. So again, the gold standard would be, however, that I'm just looking at your paper. I find your results interesting. Maybe it's a simulation study. Maybe it's like some like analysis of like GText data. Maybe it's an analysis of data I generated. No matter what it is, the ideal situation is I look at your paper. I'm like, doubts. I go to your whatever, like your whatever you're posting this, download the data, download your like pipeline, and then go from original data to final results and manuscripts like that. There's a lot of ways that you can do this, and there's a lot of reasons why you might want to do this. My personal favorite is, laziness. I hate Illustrator for burning passion, and I hate InDesign for burning passion. So the less amount of time I spend on Adobe Creative Cloud, the better my mental health is. Also, how many times have you guys had to go back into like a final figure and you have final go for real? Like, like it's really annoying, but also really dangerous. Like if you go, I didn't include any like snapshots like this of like the number of like articles like that have been corrected in Nature because, oh, we showed the wrong figure. Like the figure showing like the data 8 iterations ago. It's not right. It's like this label's wrong. Like that's something that's like, it's so easy to happen because you're manually doing it. So the more you can safeguard your data against yourself, the better, right? So there's a lot of like prescriptions on how to do this. Like I found like this one like Piper, for example, of like doing it. Our studio, like the company that owns our studio, actually has their own like recommendations for things. eLife has their own recommendations for things. So I'm going to be talking about like one particular one, which is like my particular like version using this environment in R. And I want to really stress that like this is one of the many ways you can do that, which if you think about it, is kind of meta reproducibility crisis, because then you need to know that all these things exist, which is why having things like a Docker container at the end actually makes that quite convenient. So in my workflow, and what we will be working on today, I'll use packages like renv, which just provide basically like the same idea as Conda. It's an environment manager. It will let me bookmark particular versions of particular packages, keep track of that, and it's at the end of the day one simple YAML file that I can then copy from one computer, put into another computer, and get all the things like back up and running no matter what computer I'm running it on. So for example, when Savia was down, I just had this file already on my laptop, so I just installed the packages on my laptop and I kept going. Then, this is really great for all the analysis work, but to actually see things and keep track of things, I use this workflower package, which was made by someone in my old university, which basically provides a website format with integrating with like GitHub and whatever host service you want to use, that lets you both code and track all of your code and figures in a website form that's very convenient, very interactive, and makes for a great compendium to any paper, to be honest. Obviously, I write everything in R Markdown because it's great for like, all these reproducible manuscript things. In some circles, I'm a heretic. I prefer R Markdown for Python than I do like Pi, like whatever you want, because like Jupyter, because it's a file at the end of the day. If I want to edit it in Nano, I can. And then I can like actually load it up in like something and compile it however the hell I want. But it's also supremely useful for all that like continuous integration stuff, because that is a file that any any computer anywhere with R can just compile and figure out all like the end results, right? And then, at the end of the day, we do want to get published. We don't want just to have like things in the ether. So there's actually some interesting packages like articles, which will format all of your R Markdown like end results into like a PDF that looks like an Elsevier journal article, or it looks like a like nicely formatted like I don't know, like AAAS like paper like format kind of thing. And other packages like ggpubr, which is really convenient for just like making your figure look good and not thinking more about it, or Plotly. So now is the show and tell time of like for this like talk. I'm going to show you a couple of examples of beyond manuscripts just to start. Like for example, my CV and my website are both technically made using these like fundamental principles. So it's not just publications you can apply these tools to. And then I'll go into like workflow, show you some example sites, and then kind of show you an example of like what this looks like A to Z. And then we'll go into everyone's data sets, and I will start doing a live demo here of mine. And then anyone who wants to play along, I have that data set in this flash drive. We'll take a break before then, so that way everyone can get set up, and then we'll go. Sounds good? Any questions so far? Deep existential angst? Cool. So, like I said, so everyone knows GitHub in this audience. Thank God. So this is like my GitHub repo for my CV. And so you might ask yourself, Manny, why is it that you use an R Markdown file for your CV? Like, what the hell? And it's because I'm allergic to LaTeX. So I don't really want to like work in LaTeX if I can avoid it. But also I have this like nicely provided that I found online tech like outline actually that like just kind of describes how my CV is always going to look. And I don't want to touch this with a 50-foot pole because I'm going to break something. So instead, I'm going to go over here to my R Markdown file, which describes, the fact that it's going to use that as a template. And then all of my nice header information, which doesn't change, thank God. Well, sometimes it does. And then I have like my research interest paragraph, my education, all that other stuff. I'm just going to flex my like my publication record apparently, right? So it's nice because these are just like simple like text. I don't have to think twice about it. I'm having this ongoing problem now that I'm applying for things where I keep forgetting all the things I was doing like 2 years ago. And it's been helpful that like around 2 years ago at the same time, I was starting to just like put in lines. Like every time I finish something, I would just go on, add a line, because GitHub now lets you edit things on the website and commit. So, you know, I don't have to think twice about this, right? So I have all this stuff, and then at the end of the day, I can just knit it, like with knitter on R, and it looks like this. So it has like the nice like things. It looks sleek. It's like I was never going to like do this much work in real life. Let's be honest. So the fact that all of this is automated, so I don't have weird formatting errors because I accidentally put a curly bracket in the wrong place or like something weird, right? It's nice. And then also it's on GitHub, so everyone immediately like goes to it, right? And they can find it. Every time someone sends me a like, hey Manny, can you send me your CV? I literally just go over here and I send them like, okay, yeah, sure, this is like the most recent copy of my CV, right? Like here's a link. So it's like really convenient at the end of the day. And so like, you know, it's on GitHub. If you guys want to see what this looks like, just go to github.manny.cv and then you can find it. You can fork it. Before I go into, I'm gonna actually talk about workflow before I go into my website real quick. So this is kind of like the homepage for like the workflow website. And essentially, all this is like a front end for a lot of like the otherwise annoying work that you would have to do, but there are some tools and some like aspects about Workflower that make it really useful for data analysis, specifically if you're doing things like simulation research or you're doing things like making a lot of figures. So in general, the way that it works is you will just have your R Markdown, like your your environment, probably RStudio, realistically speaking, and the Git repository to like host everything at the end of the day, and then everything else comes on top. At the core, that's just it. So it's as simple as you just run like Workflower, you load it, and then you do workflow start, and that creates this outline in like whatever folder you told it to like create it. It creates this like folder directory of files. This is beautiful. Every time that you are mentoring a student from now on, you will tell them just type in Workflower start, and you will never not know where their data files are, right? It's like it has like a data folder. This really helps a lot with like the habit of like, I'm just gonna have like a scratch like space in like my home directory, and like it's gonna be like all these millions of files. Like it, by having this format, you do end up following it. So like in my personal case, I'll have the data, the experiment types, the replicates, all that fun stuff. And then let's say I have code that I just use to like process the hell out of everything. That will, and all of that output goes into the output folder. And then I can symlink it back to the data folder. So that way I keep this like flow of like data to output to data to output. And that way I always know if I want a final thing, it's going to be in the data folder. If I want chicken scratch, it's going to be in my output folder. And then the analysis is like the actual, like if we think back about that pipeline from like data to results to like the analysis. Analysis is actually where like the brain power is like mostly happening. All the markdown files that are actually like making things and making figures that then when you knit them in the workflow environment get put into the docs. And the docs is where then all the hosting and all the like magic releases start happening. So it's easier for me to show you than now an example of one of them. So this is one of the authors, like they were studying this like bike share program in Chicago using this. So you can see that there's a couple of interesting things. First off, this is an actual website. Like, you know, this is like the header like area. If I click here, it just brings me back here. I can go into like the setup page, all that fun stuff. So you don't at any point in time have to like design the website. It's all like being handled behind the scenes. And then there's this like workflow checkbox. And this is actually like the big thing about like the package. If you click on this, it gives you a reproducibility checklist. So when you see that green check mark, you know that you haven't messed up somehow. You can just kind of keep going and living your life. But this tells you, you know, when was the last time that this file was updated? What checks did it pass and what checks did it fail? And what directory was it knit in compared to like the main directory, right? So if I go into what those checks mean, you know, so there's like a check to make sure that the R Markdown file is up to date. So when it says up to date, it means actually has the R Markdown file been committed to GitHub. So if this has a red X on it, it means that you haven't Git committed like your file yet, and it's not because it's not Git committed. you are at risk of losing whatever the hell is going on here, right? Or more importantly, you don't have any version tracked. So if you broke something between your last version and whenever you committed next, you have no way of finding out when you broke it, right? And then it actually tells you what is like the head string for all the GitHub, for like the Git repository you're in. And then the beautiful part is that it also tells you it summarizes all the previous commits and the previous commit messages that used this file. So for example, this is just the home index file. And these are all the different commits that he did, like for all these things and all the descriptions, right? It's about as good as you put into it. So like in this case, right, like not terribly like informative, but informative enough that I know what happened between these two commits, right? And then I can always just like hide it again. It's quite nice. So if you then go into like what it looks like for the data analysis portion, right? It's just like any other like R like thing. It'll have like your nice table of contents here for you to navigate. It still has this reproducibility checklist at the beginning, so that way you can like go through things. You'll see that when it's not like the home screen, when it's actually like an analysis, there's many more checks that get done. And those are things like, for example, making sure that your environment was empty before it ran the git commit. This is something that, you know, it's just kind of like, it should be empty. But like, how many times have you guys like run like some code and then like restart the R environment and actually the way you ran the code was out of order? So like you were using something you generated later to generate something earlier. So because it tells you an environment is empty, you know that this will run start to finish. It sets the seed for you from the get-go, so all of your like simulation studies will be reproducible right off the bat. And it has like a file in the root folder that it makes that you can actually change the seed to whatever you want. You know, it reports the session information towards the end. It tells you if there's no caches, you're not using like the R markdown like cache option, which, you know, sometimes you may actually want to do that. Say it's a simulation that's going to take like 10 years. You don't want to rerun that every single time that you actually like git commit this. So you can like tell it, don't rerun all the cached like environments, just use those. But it will warn you are using a cached environment, right? So if you're changing something in the code and expecting this to change, it won't happen, right? And likewise, like relative paths, you're not kind of like letting everyone know what your username is on your desktop and like other like key information about your desktop and repository version, right? So it's nice because it has like all this like formatting, so it's all that fun stuff. The session information at the end is kind of like the basic like stuff, so I just know like what was used to like run that analysis. And this is what it looks like, just the bare bones, right? So if you then look at what that looks like for, let's say, okay, that's what it looks like for bare bones. So it's on it's a website though, right? So like I'm currently looking at it on websites. There's a million hosts that services that you can use. I use Netlify because it's free, which is a great reason to do anything in life. So basically all this does is that it will take, for example, from my GitHub repository, it will then proceed to just publish everything that's in the docs folder without like thinking any harder about it. But also, if you wanted it to actually regenerate all of your data, regenerate all your analyses, you can tell it to use a script to build your commands, your website, in which case that's how you would integrate that continuous development environment aspect of things. If I show you what this looks like, for example, for my websites, this is all made in R. If you want to see how I made this, it's just using like Hugo and Blockdown, which are two more like R packages, so I don't have to think about HTML in my life, which is great. But then I do use like my R Studio stuff the workflow stuff for some of the posts I do. So I can continuously struggle on this. So in a previous committed version, I used to actually have like, I literally just copy and pasted the function that it uses to generate that like check to like add it here. And you can see that, you know, you can have this like website, everything's in R. You can just generate all your things like right here. But the beauty is that it lets you do some other more complicated things, like for example, have this graph, which is an interactive version of a graph, right? So for many of my analyses, I don't want to have to like stare at the abyss and then just have the abyss stare back at me. I actually want to look at things. So this is like a graph, for example, of all the mammalian data that we used to have in 2014 for body size and lifespan. And I just want to look to see what are some interesting species for me to study for the rest of my life, right? Because there's this general positive correlation that's great, but as kind of was introduced, I study evolution of aging, and I would like to find out why, like for example, the bowhead whale all the way over here is like, lives like 100 and, sorry, blue whale, the bowhead whale is like this one, that like lives 211 years, right? I want to find out like why it lives 211 years and see, can we use that for human health inferences? But what I don't want to end up with is a Frankenstein's monster of like a bowhead whale-sized human, right? So in order to do that, it would be nice to look at a clade that doesn't actually have that like positive like confounding factor. And so you can see kind of sort of that the bats are kind of coming off that line. And you can double click on like, any one point, or I can double click here. So only look at the bats. I didn't have to do any code. It's all interactive, right? It's nice, beautiful. And more importantly, my advisor was not a comp person. And so he was able to do this as well and verify all of my results, which was great. So you can see that bats are really cool. They live much longer than you expect, given this regression. And there's this like clade of bats that actually is like a vertical line off of this, which I show again later here. This like one group of bats just like lives, their body size is independent, their lifespan is independent body size, making a really interesting thing to me to do my postdoc work, right? But this is also another interactive graph. So I did like the individual regressions. You can see that there's like weak positive correlations, right? But there's like some of these that actually have a very strong positive correlation. So again, really cool things, ways for you to like look at your data. And when it comes to actually brass tacks, right, like what you want is to eventually this become a publication. So when I got into this in grad school, I actually like went from start to finish in like making a publication based on these reproducible manuscript like tactics. So you can see here that this is the same kind of like structure that like workflow enforces, except I made a new publication directory, just so that way I knew like, okay, where the final like everything was. And it's very nice and stuff, but I also provide all the instructions on how to reproduce all of this. So this isn't fully reproducible. This gets like a silver star, doesn't get a gold star. You still have to like install all this stuff and install some stuff that didn't used to be on Conda and then clone the repo, but I give detailed instructions on every step of the way. And then I give you the citation and the link for all the genomes I used and all that other fun stuff, right? But if you wanted to, you can actually just use snakemake here to like generate this and then generate this and then generate this. And then eventually you get to the point where you can use snakemake to just generate the entire manuscript. So once you set everything up, for example, I could update this nowadays and then put all this on like a Docker environment, and then everything would be like fully reproducible, right? But the nice thing is that then I have like, like my manuscript R Markdown file, and I also had it, no one likes to like edit PDFs or R Markdowns for like editing. So it actually knits into like a docx. There's an entire, I forgot to include this, there's an entire like package in R that's like lets you knit into a docx, but then it reads that docx back into an R Markdown because it just uses like behind the scenes editing things in the Word document. So I could send my advisor the docx and he could make the edits, and then I could load it back into my R Markdown. And in that way, we could keep collaborating, even though like I'm allergic to a lot of other software. It has the PDF stuff. It has log files when something fails. It has this HTML file. So like everything else, there is a website version of this paper, which I was using to like actively like edit things, see things, how did it look, all that fun stuff, right? So I didn't have to do anything with any crazy citation manager. I literally just used like R's like built-in citation manager. And I just have like a bit text file that's also part of that, right? So like all that's handled for me. You'll notice that there isn't a real like format here. Like it's just kind of like text. For that, you would need to actually use the articles package. So the RStudio people have like this articles thing, which is just a way to basically like use a template from like one of many like publications. So like, let's say, for example, you're submitting things to Elsevier, and Elsevier used to be much more like insane about how they wanted like everything to be formatted before they read everything. So you could spend an entire night like crying on your computer trying to understand why anyone wants something in such a weird format. Or you could just tell it that tell R Markdown that you wanted that really weird format that someone made in LaTeX, right? And so just like I make my CV by just using reading in a LaTeX template and then spitting out like real like things, this actually lets you for many different societies just spit out like, for example, if you want to go to like, I don't know, where is it here? There's like some here conferences like here. So if you want to submit a conference paper, for example, like there it is. There's like a like Arvix like templates here that someone like put that I've seen a lot of people use for bioarchive as well. Like I said, there's an Elsevier and Frontiers like version of things. So in my, actually let's see if I kept it all the way to the end. So like this is just like the basic one. No, this is the, you see how this is like preprint submitted to Elsevier? So like this is using the Elsevier like template. So if I go back to my R Markdown file, in the outputs, I have here like the book down PDF book, and that's because I wanted to have like links, just other things. But the base format is using the article's Elsevier article. So as a result, then I can just have this nice like format. It's exactly what they were looking for. I don't get any like points off for like not like sticking to their formatting methods, and I end up with like, you know, an Elsevier format article. So all these tools together are really useful for like kind of saving your time from like all the stupid death by 1,000 cuts of academia, right? So like all like, and that's really all reproducibility is. It's just trying to like save your future self from your present self, right? And so the nice thing is that, you know, I went through a million like revisions of this, like everyone does, right? But at the end of the day, like this ended up on eLife, and the nice thing is that every time someone came in for revision, I didn't have to worry about whether or not like text I had, for example, results. Some of this text, for example, you see how I'm like, oh yeah, like this is really cool. They have like this weight and this confidence interval and this is the range and all that stuff. If I actually go back to like the R Markdown file, like I have like all these chunks that's just making figures and just like reading things. Nowadays, I would probably save these in a different file and then load them into it using RStudio. But for some reason, I wanted a monolith, so I got a monolith. But somewhere here, I'm actually using like the R inline code. So for example, this is how I would like cite the figure, and that's how the figure ends up getting like bold and stuff. So notice how I'm not mentioning the figure number anywhere. So figure 1 is not referring to anything weird. It's literally referring to the intro figure, which I have labeled, right? And then like, you know, my citations are the cite keys from like the things, and like there's like the genomes and stuff. Here's like my methods. So yeah, anyways, there's like a way in R, like, right, to like just have like the, like, yeah.
Not to rush it, but I think we are 10 minutes from roughly the end of the hour.
Yeah. Time flies. Anyways, you have like your R Markdown like thing. You can like put R code in there that's actually calling the numbers. So every time something changes its analysis, I never have to worry about it. Like this thing was changing alongside it. So I never ended up with like those weird like issues that like you were talking about something random that's not real, right? So with all that in mind, I'm actually going to show you like what it looks like right now for one of my projects, right? So this is like my project for, actually I'm going to show you a simpler project. I'll show you someone else's project. So let's do this one. So essentially, like if you have this like whole like workflow thing, actually, let me show you in the interest of time, like the final product. So this is what it looks like for an analysis I was doing on DNA damage response in QTLs. So this is like the websites that ended up with workflow. You can see that I have not been like good. Specifically, this is I did not like commit the changes yet. So this is all stuff that I have made changes to recently and they were uncommitted. I did my different analyses here. Here's like one for like the etoposide dose response curves. And there's a lot of analyses here. So it's actually quite easy for me to just go over here and then like show you like all these like fun graphs, right? And I can go over here to like the PCA analysis and all these like different plots. It works out quite nicely, especially when it's something like, for example, doing RNA-seq, because you can use the EBSeq package like straight up in here, right? And then I find out all like the millions of things that like could be silenced, and then all of my nice upset plots, everything in R, right? And again, I'll go through this. Unfortunately, I don't have time for this, but all this stuff is in like the GitHub repos that you can find if you want an example. And the vignettes for like workflower is actually like quite useful in of itself to get started. I think the most beautiful part about this is that it requires relatively little like extra work on your front to change the way that you're doing things. It just really requires you to like use a couple of extra packages on top of what you're probably already doing if you use R. If you use Python, you can still use actually all this stuff. You just do need to use the R markdown for like workflower, but you can call R, for example, in like the chunks for like an R markdown file. Sorry, you can call Python from the chunks of an R markdown file, for example. So yeah, hopefully this has been helpful in some way for all of you guys, and hopefully you do like end up using. I didn't get to show some of the nice things, but you know, that's it. Questions, comments, concerns?
I have a question about how you use like SnakeMake and Workflower. Like, which parts do you use?
So yeah, it's a great question. So I use SnakeMake at the beginning. So in terms of that like workflow, like of how you make a paper, right? I'll use SnakeMake to like actually generate all the data files that then I will be analyzing using Workflower. So then you can have an R script that calls Workflower and runs everything for you, which is what I did for like the manuscripts, like the R markdown file. So I just had like a small R script that just called Workflower's like build tool, and so it just went from everything from scratch in the directory and built it all up. And Workflower, thankfully, is smart enough that if you're making a reference to R markdown file A and B, it will knit R markdown file A before it knits B. So that's nice. Other questions? Well, there's a class here, I assume, afterwards. There's like 5 minutes.
We have enough time. Yeah, we don't have to rush out.
Yeah, so I can show you them.
I was just saying that the seminar technically ends at 1, so I think who has to go, no shame, you have to go. But we also have the room until 1:30. I don't think we should rush out.
So I have this like a file here, for example, like for the longevity quotient stuff that I do. And this is like just like a deep dive data analysis, right? So I have all like my fun things here that I'm like looking at. And then this is really like the point, for example, where I'm like showing like, how little I have to care about, how little I have to worry about when it comes to like, like making an edit. Like, that's not a bad looking figure, right? It's like, it's fine, whatever. These are the only two functions I'm actually calling to make that figure look like that, which is like really nice, right? And so these are from the ggpubr package. And so it's just kind of like has a bunch of like different tools and stuff to actually like make graphs that look just like this from the get-go without having to like think any harder about it, right? Like that is just like, for example, this graph here is just this one like function like right here. So if you're doing like, like ab initio like kinds of analyses, like you're doing it from scratch, you may as well use their stuff. But if it's just like, your own like custom, like special snowflake kind of graphs, right, like this, then you can just kind of add like these two functions to make it look nice at least. I think my advisor is extremely like picky about like the kinds of graphs that he wants, and he has never had any issue with any of the graphs made by like by ggpubr. So saves you more time as well by like going back and forth with your advisor. So as
Can I
an
ask a question? How well is this for like collaborative projects?
extremely well, actually. So the whole point of Workflower was to force collaborators to play nicely and within the rules, right? So everything's on GitHub. it depends on how allergic your collaborators are to like GitHub, right? If they're the kind of people that keep sending you things and like they're not like really like, it's just like random emails of your files, you can so long as you have the file, right? Like that is like being tracked in some way, shape, or form, you can always just handle the commits yourself, right? And do the tracking, things like that. The nice thing about it though is that if everyone is using is using GitHub and they're pulling everything, right? And they're pushing everything back into GitHub, this is going to keep track of the different changes that different people do. So then if someone breaks the analysis, you know what commit it was that broke the analysis, which is like really convenient. The other thing is that sometimes you'll go back and forth. I think I do have an example of that here. Sometimes people will go back and forth about how they want a graph to look. No, I don't have an example here. Yeah, so sometimes people have like an example that's of like how they want a graph to look, and then like you make a change and they're like, no, I don't like that. You could undo all the changes, or when you've made several commits to a file, there's usually like a little bar here that says past versions, and you can actually go and scroll through all the previous versions of said graph, which is really useful when you're all like talking together about like which variant of this graph like made sense, right? And like I said, the other nice thing is that because R has the ability to knit to a Word doc, when you get to the actual like writing portion, you can just send everyone a Word doc to make those changes. Where your mileage may vary is if everyone likes Google Docs, and there I'm like,
I was gonna say sending people Word docs does not sound.
Yeah. So the nice thing is that when you send people Word docs and they send you back the Word doc, right? Like then you can just, so if it's like you're sending it to your advisor and your advisor sends you back edits, and so you make the like changes, like you can reload that Word doc as the R Markdown file and then just keep going from there, right? So that's really nice and convenient. When everyone has to collaboratively start writing on things, my experience of like Google Docs was that if you kept the Google Docs with like the writing, and that's like completely fine. Sometimes it breaks the behind the scenes like formatting things. If you got like way too crazy and creative with your like R Markdown file, like using way too much esoteric packages, that could break things, but generally it was fine in my experience. Again, like that's I think where like one thing is like the growing pains. Like it does require people to edit things in a like particular way and like not go too crazy with changing the structure. I like that question a lot, actually. Thank you. So like as an example of like everything together, right? Oh, I never mentioned Plotly. So all those interactive plots I'm like showing you comes from Plotly. It's like a service in of itself. It's actually a JavaScript graphing library service. So it's agnostic in real life about like how you want to like do things. So there's an R package version of this. There's also a Python version. There's like a Julia version. But the nice thing is that they have all these examples of how you can make things. And they have a function called ggplotly that just takes a ggplot as a function and then spits out like one of these interactive graphs that I've been showing you all this time. But you can then add on to that with any of their stuff if you really want to go crazy.
Oh, we are at 1 p.m. So thanks everybody for attending. Thank you, Manny, for the great.